Okay, so today we are finishing up the second part of protein synthesis. We're looking at that second step, which is called translation. Big ideas, we're just continuing on from yesterday, making sure that you understand what is the central dogma, be able to understand in that process of the central dogma what it is that makes the RNA, and now really looking at what it is, words, that make up the proteins. And in doing all of this, hopefully you're able to now look at the structure of DNA and RNA and then also compare them to proteins. So again, as we're looking at the central dogma, we already know DNA is used to code for RNA. That RNA is used to code for proteins. Kind of rephrasing that. DNA we're going to think of as the master blueprint. It contains all of the information to make you, and every single cell in your body has a complete complement of all of the DNA that you need. All of the instructions are there in what is called your genome. So that genome is safely housed in the nucleus, and it's just kind of sitting there not really doing much. But it is being photocopied. That process that we looked at yesterday where we take the DNA and transcribe it into RNA, that's really just kind of photocopying it, right? So in that process, we're just taking one type of nucleic acid and converting it into another. We're just talking in the language of nucleotides right now. But when it comes to proteins, we're going to start switching things up a little bit. So let's take a look at what are the subunits of protein. So remember that proteins are just big old strands of amino acids, and amino acids are going to look something like this. We've got that carbon in the center. Four things are going to be attached to that central carbon. Those are the carboxylic acid, that COOH, the amino acid, the NH2, and the hydrogen. Nothing super special going on with any of that because every single amino acid has that same basic structure. So when we looked at the nucleotides, it was the phosphate, the 5-carbon sugar, and the nitrogenous base, and what made each one really special was that nitrogenous base. Well, now when we're looking at the amino acids, what's going to make each one special is the R group. Remember, we did this in like week 3. Doesn't that seem like forever ago? Oh my goodness. Anyway, so that R group. Well, the R group can be really huge or really tiny. It can be kind of acidic. It can be kind of basic. It can be negatively charged or positively charged. Lots of different characteristics there. And it is that R group that is going to dictate the characteristics of the entire amino acid. So all of this stuff down here, it's like nifty, but it's not as cool as the R group. The R group really gives it the characteristics, right? So we've got an amino acid, and these amino acids are going to be strung together, pow, 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 in a specific order so that you get a whole bunch of R groups, and those R groups will begin to interact with each other, fold up, and it is the folding that matters, because what matters about a protein is a three-dimensional shape. Remember any of this? I hope so. Anyway, I know it seems like it was forever ago because it kind of was. So we've got this three-dimensional shape that is going to come together and give the protein its function. Why am I going over all of this? Keep in mind that those proteins have so many different functions. We've seen a few of them. Carrying oxygen, for example, your hemoglobin. The proteins in the membrane that are either going to allow things to get in or get out. Antibodies, really important right now. With your immune system, antibodies are proteins that recognize and bind to different pathogens, like the COVID. But proteins are also your enzymes, and enzymes have a ton of different functions. So at the end of the day, the instructions are the DNA, but what makes you is what proteins you have in you. Those are going to determine all of your characteristics. Kind of like if you picture a city skyline, what do you imagine? It's not the blueprint that was used to make the skyscrapers, it is the skyscrapers. So as we're looking back at that central dogma, the DNA to RNA, that's all about those nitrogenous bases. But now we're going to switch languages. I said nitrogenous bases, I didn't mean that. Nucleotides. But we're going to switch language now. Instead of talking about nucleic acids, we're switching over to proteins. We're switching over to amino acids. And so those are the final products that are getting made right there. And that process, changing languages, is called, there's the button, translation. Doesn't that make sense? Like changing language, translate, whatever. Anyway, so review here. We've got the DNA is transcribed into RNA. That piece of RNA isn't the entire genome. It's just the instructions for the particular piece of protein that you're making. In other words, it's just a single gene. So your genome is all of the genes that you have. It is all of your DNA. But when you go to make a protein, you're just going to photocopy the segment of RNA that contains the information to make that particular protein. So during transcription, the RNA is made. Then the RNA is going to be used to make the protein. This is going to occur on the ribosomes. Remember that your ribosomes are what are building your proteins. And because proteins are super duper important, all cells, whether they're eukaryotic or prokaryotic, have ribosomes. Boom. Oh my goodness. It's like everything comes back to what we learned at the beginning. So even though you're missing half of biology, you got the important piece down, right? So those proteins can be modified, sorted, packaged, etc. on the ER, the Golgi, into vesicles, into the vacuole, sent out of the cell, exocytosed. But ultimately right now we're just really worried about ribosomes. Okay, so let's look at the process. We've got our molecule of RNA right here. How do you know it's RNA? Well, there's some A's and G's and C's, so we know it's some kind of nucleic acid, but then it's got U's, uracil, so we know it can't be DNA. In addition, it is single-stranded. And so this RNA is going to yeet on out of the nucleus where it was made and then land in the cytoplasm where it is going to bond to a ribosome. Hello, ribosome. And that ribosome is going to start chugging along and scanning through the order of nucleotides. So remember we talked last week about the most important thing about DNA, and therefore RNA, is the sequence of nucleotides, the sequence of base pairs or nitrogenous bases. And that's going to come into play now because this ribosome is going to start scanning and it is reading the nucleotides, the nitrogenous bases, the A's, C's, U's, G's, kind of like we would read a book, right? We read the letters and get meaning from it. The ribosome is going to scan through and read and get meaning out of it. And it's going to scan through until it sees AUG, which is called a start codon. Start codon does exactly that. It says, start translating. From there, we're going to read in sections of 3. So we read words that have a certain number of letters in them. The ribosome just so happens to only be able to read in groups of 3, and each group of 3 is going to be called a codon. Let's wrap all of that up and make sure we're making some sense here, okay? So the DNA is transcribed into RNA. That's our first part of the central dogma. That's what we talked about yesterday. The RNA is going to be the copy, and that is what is accessible to the ribosome. Because the DNA is safely housed in the nucleus, the ribosomes are in the cytoplasm, and so we need a way to get that message out. That's going to be our RNA. And so the ribosomes are going to read through this RNA, and they're going to read it 3 bases at a time. Each base is called a codon. Each codon says which amino acid is going to be used. So 3 base pairs codes for 1 codon, which codes for 1 amino acid in the protein sequence. Okay, we're always going to start it at a start codon and stop at a stop. Not too difficult there, and I think it's helpful to take a quick little peek at that process. Okay, so what we've got right here is the RNA. Each of these different colors is going to represent a different codon. So 3 base pairs, or not really base pairs, 3 bases in the RNA. Reason it's not a base pair is because RNA is single-stranded. So right here, for example, you've got the AUG, the start codon, and that's going to signal to this little blue blob right here, which is the ribosome, that this is where we want to start reading. This is where translation needs to begin, where we're going to start making the protein. So this little guy, the squiggly guy, you don't need to worry too much about the details here, but this is also a type of RNA. It's a type of RNA that kind of works like a worker, bringing the materials to the work site so that we can build this skyscraper, build this protein. So in this case, he's bringing the particular amino acid that is coded for by the RNA. So the RNA says what the building material is, and then the ribosome sends for it, basically, right? We're really simplifying here. And then the correct amino acid starts getting brought into the ribosome and string together kind of piece by piece so that we can make the protein. So some words, some things are happening, and now here's a new little worker. It's going to recognize what the RNA says. So this particular sequence of bases is going to indicate which amino acid needs to get brought into the ribosome to be able to build the protein. And so this process is just going to continue with new little workers coming in, bringing their amino acid, and the amino acids that have already been brought in are getting added and added and added until eventually what you have is a finished polypeptide. Polypeptide meaning string of amino acids. Eventually we get to the stop codon, and that's going to allow the whole thing to fold in and produce your finalized protein. Trust me. I mean, I know it's a spoiler alert because we're not there yet, but it's gonna happen because I know these things. Oh, look. Okay, so there's our stop codon, and that's like, hey, everybody just break up. Break this party up. Let's socially distance. Oh, too soon. And this sequence of amino acid is our polypeptide, and that can then bloop, fold up into a functional protein. Good story. Also, if you're ever wondering, the NDSU virtual cell, fantastic videos, really, really good. So anyway, that's my little side story. Let's go back to this guy right here. So hopefully that made a little bit of sense. Let's kind of quickly recap. DNA is used to make RNA, is made used to make proteins. We've seen this. So in the case of DNA and RNA, we are looking at nitrogenous bases of A, T, C, and G, or A, U, C, and G, as what really matters as the language of the nucleic acids. When we switch over to the proteins, we're looking at amino acids, and there are 20 different amino acids. So there's only 4 different bases in DNA, 4 in RNA, but 20 in the proteins. So let's go ahead and take a look at how to go from RNA to proteins. Luckily for you, scientists have figured this all out, put it in a handy dandy chart so you don't have to worry so much about trying to figure this whole jaunt out. So the way that this works is you're going to use whatever the codon is. Remember that sequence of 3 bases, and from that you're going to be able to determine which amino acid is going to be used in that particular spot. Okay, so for example, let's say that the RNA says A, U, G. Okay, what we're going to do is we're going to go over here to where it says first base to the A. That means that whatever amino acid we're looking at is going to be in this row right here. So we're starting with A, and then we're going to go over to the column that has the header of U, which means we're going to be in this column and this row. So somewhere in this box, A, U, our last one is G, which means we're going to come over here to the third base, look at the G, go over, and that is MET. Okay, a thing about amino acids that you should know. Because scientists are kind of jerks, they made a whole bunch of different ways to refer to each amino acid. So 28 different amino acids, we can refer to them by their full name, their abbreviation, or a single letter abbreviation. You're welcome. In this case, we're looking at MET, which is methionine, which is abbreviated M. You don't have to memorize any of that. I will give you this chart. You will have it in front of you for any work that you need to do. But big picture, just make sure you're able to understand that DNA codes for RNA, that RNA is going to code for proteins, and you're going to use this chart right here to be able to figure out which set of 3 nucleotides codes for one amino acid. So AUG, I said that is the start codon. So we always are going to start our protein sequences with the AUG, with that start codon, and AUG just happens to code for methionine or MET. Okay, so I'm going to give you a couple. I'm going to say them pretty quickly, but I would recommend that you take a moment and hit pause and think it through and see if you can figure it out, and then hit play as I'm going through this. So let's say, for example, I've got CAC. To figure that out, we're going to go C over to A, this column here, over to the C is his or histidine. Let's say I've got GCA, I'd go G over to C. All of these are alanine, just so that you can see which one it is. It would be the third one down. But oops, my bad. I got a little excited there and exited out of where I needed to be. Anyway, it would be the third one down, this alanine here, but they're all the same. As it turns out, there's some redundancy, and that's fine. Let's say you've got UGG. Ugh, right? We're all feeling that. U over to G, down to G. We're looking at TRP, which is tryptophan. One more for you. How about UAA? Okay, that's a stop codon, and that would say you've reached the end of your gene. The polypeptide is complete. The ribosome, the RNA can split apart, and what you're left with is a polypeptide that can now fold up. As you just saw, because I hit the wrong button, there is another way, another chart that you can use to do the exact same thing. In this case, this has kind of that radial view. So we're going to start with the inner circle here for the first codon. Second codon is the second circle. Third codon is the third one. Same thing, make sure you know how to read these. So let's say, for example, I've got AUG. Just like before, we know that's our start codon. We also know that's methionine, and we'd find that by going from A out to U, out to G, and here's that methionine. How about if I've got CUA? I'd go C, U, A, that's leucine. So the full name is leucine, the 3-letter abbreviation is LEU, the 1-letter abbreviation is L, and that is going to come into play for a couple of our activities this week. So I will post both of these charts, but the single letter abbreviation is what's going to come into play. Let's do 2 more, and then your assignment is simply going to be, not simply, it's not always super easy, but to take RNA sequence and translate it into a polypeptide. Okay, so if I do UGG, U to G, there's our tryptophan, the abbreviation for which is W. I don't know, science. GUU, G to U to U, that's valine. How about if I do GAA? Okay, G to A, glutamic acid, abbreviated E, because science. Good news, if you are thinking about going into medicine or biochemistry or biology even, you will have to take biochemistry and you get to memorize these. So anyway, on that note, for all of you people that are not looking at majoring in biology, just, be grateful that you need to know this for, I don't know, like a week, 2 weeks, then you can forget it and just move on with your lives. With that, be good. If you've got questions, let me know, and I will chat with you continually through YouTube. Good times.